So second talk in the early detection session is from Guido Verbeck from University of North Texas. And Guido has a very early start today. I think it was 3 or 4 a.m. So his talk title is The Determination of Breath Biomarkers and Metabolites for Disease and Health Constituents using Non-Invasive Direct Inject Mass Spec. Guido is a professor of chemistry and biochemistry and an expert in mass spectrometry, especially instrumentation design and development, and the director of the lab for imaging mass spec. He spent over 20 years developing mass spectrometers and was awarded the Young Investigator Award from the Air Force of Scientific Research. He's currently developing mass specs for single-cell analysis for cancer biomarkers and nanoparticle development, pharmaceutical and toxicological biochemical effects. Welcome, Guido. We look forward to hearing your talk.
Hello, my name is Guido Verbeck. I'm a professor of chemistry and biochemistry at the University of North Texas. And I've been in the mass spec community for now going on over 22 years. And today I'm going to talk about using direct inject techniques to help us identify some of the VOCs and small molecule markers that could potentially help us in breath biopsy to determine different types of disease states. And we do this with direct inject mass spectrometry because we want to see full gas loads without separations front end to see if mass spec can determine these different types of markers. We use an LTQ mass spectrometer for these types of instruments, and we also have an LTQ attached to an Orbitrap, which is a very high resolution mass spec. And we can look at both of those for identifying it, but the low resolution mass spec is a better one for us because this is going to mimic what the fieldable mass spectrometers look like today with roughly about 1 AMU resolution with quadrupole mass spectrometers. Direct inject mass spec we'll start with first is just the regular nano extraction. So if we can look at a cell line, see a difference between a tumor state and a healthy state or a disease state and a healthy state, we can use this technique to extract and then start going to look for chemistries that we're interested in using for biomarkers. So we wrote a review paper at the true one cell limit because a single cell has a myriad of meanings, and we wanted to make sure that we were conveying that we're actually doing mass spectrometry at the 1 cell limit and doing exactly the chemistry that we're interested in. And so here's a video of using the direct nano extraction on a cell. So if you see a cell, it's going to be apoptotic or there's a problem, we can do cellular analysis or an organelle analysis. And in this case, we're going to laser breach the cell, which you can't really see under the microscope, and then we're going to extract the adipocyte lipid body organelle, one organelle out of it, and then we're going to spray that in the mass spectrometer. So truly a 1 cell operation. And then here are the responses that we get. So you can see that in these cases, this was a 3T3 line that we differentiated to make the fat cells, white fat cells in the rat. And so you can see right here that we have the triglyceride distribution here, and then we have a high resolution Orbitrap. And so we can actually monitor the changes in these triglyceride formation to see if we can use fatty acids or the lipids to be able to determine the disease state. So we did apply that to breast tissue for a cancer patient, and both of the tissues were harvested at the same time so we can account for diet. And there's a healthy tissue on the left-hand side and the tumor tissue there on the right side, and we did the extraction that I just showed you before on these cell lines. And repeatedly, over and over again on these cells, we got a knockdown of the 54 acyl group. And when we did MS on that, we found out that there was arguments in the literature about linoleic or oleic acid that was being consumed during breast cancer, and we were actually able to show that the linoleic acid was the one that was being knocked down. And this was reproducible. So, of course, we just didn't do this experiment once. We did it multiple times, but then we also did it on multiple patients where we also got other breast and healthy tissue from them, and we continually saw the knockdown in the 54A. So we published that in the Journal of Analytical Oncology a few years ago. So that's one way to extract it and look at it. In this case, it's fatty acids. And so fatty acids are interesting looking at. Pyrolysis, fatty acids and pyrolysis coupled to mass spec can also be something that we could do in the breath biopsy world. Teslin was the other way. So paper spray is a way to also look at the VOCs, because you can take something that's got a heavy matrix. Like in this case, we're actually going to take the respiratory cells from the normal scrape like they do for the PCR test, put them on the Teslin lysed, and then be able to spray them and look for some VOC differences between, for instance, a person who has COVID or a person who doesn't have COVID. These markers were identified in 2016 for the flu, SARS-1, MERS, and so we applied that sort of same idea to COVID when we got the cells lysed to see if we could see these aldehydes and ketones that were showing up on SPEMI, solid phase microextraction coupled to GCMS, and see if we could identify those also with direct inject mass spec, which is much faster. It's one way to do it. But of course, we want to train the breath biopsy world on what these compounds are. So just to kind of introduce you to direct inject paper spray. It's paper, cellulose. In our case, we use Teslin, a material called Teslin, because it is essentially a printable glass. And so its pore structure is much more controllable, and it allows us to spray chemistry of interesting things that are especially more hydrophobic. And we get better reproducibility with this material, so I won't spend much time talking about that. But one of the areas we wanted to start with, of course, is the fentanyls and the cannabinoids, and being able to identify those, the metabolites on the breath, to see if we can see these. And so you can see here the fentanyl compounds and the ability to be able to look at the different analogs, because we're interested specifically in the volatile organic compounds that cross the respiratory blood tract membrane, and be able to determine whether a person is under the influence of certain drugs, especially fentanyl in this case, and then we can tell all the analogs. So here we've done a full analog sweep. By this point now we've done 240 fentanyl analogs that we can identify and created a library for, specifically for using breath markers in breath biopsy. Here's just another example of each one of those experiments. So we took that Teslin, that same material, and we applied that to COVID-19. So we had some samples of lysed cells with COVID-19 and lysed cells without COVID-19. And we can also do this on a controlled line that's called 223E, and try to determine when the cells go apoptotic, is there a specific type of VOC markers that we can see, and then can we train the instruments on it? So we published this paper in the Analyst in 2020. And so the idea is that this life cycle of SARS-CoV-2, the potential lipid modification is shown here, with a modification of the fatty acids and the cholesterol synthesis. So this happens in the membrane. When the cell goes apoptotic, it releases all of these compounds. And we want to be able to determine whether or not these organic compounds are coming off, and can we tell the difference? So one of the big differences that you can see here, just from this quick picture, and I'll show you some more in a little bit, is the COVID negative versus the COVID positive samples. The organic load is much higher for the COVID positive samples, which are going positive there, and the COVID negative going negative, but you can see just a huge amount of organic signature that's coming out, which was promising to us, but we needed to make sure that we could see compounds that were reproducible and the compounds that were volatile. And we want to keep these low molecules that we're interested in, because if we're doing breath biopsy, these higher markers are great for if we wanted to do a swab test and using Teslin to look at it and lyse the cells, but that's a lengthy process. But we're interested in more of these small molecule compounds down here and seeing some reproducibility in them to do breath biopsy. So here's our process. The cells were swabbed and they were done with PCR and lysed. before they were doing PCR, and then they were sent to us lysed and dormant, no active cells in this case. And so you can still see, even in the low mass realm here, this is 100 to 350, that there are some differences and that there's much higher organic load. So we went through to start identifying what these compounds were, and we started finding, one, that we have some aromatic compounds, which are good, but two, that we're having some aldehydes and ketones that were being present, which points the same types of compounds that were shown in the paper in 2016. And so here, when we started to pick out of that group of cells that you saw from the beginning, which ones were positive and which ones negative, we had a clear delineation. We have one false positive and one false negative in that set, which is pretty good compared to the fact that we're using PCR as the gold standard. So that has some difficulties we all know of trying to use PCR as our gold standard to prove our techniques. But we thought, hey, this is pretty successful and pretty reproducible. So let me show you exactly what that looks like. Here's 4 different positive samples. Really high organic load on the respiratory cells. And then here are the COVID negative samples. And so you can see that there's just a really, really large difference. Greater than 10%. Here were the markers there, and then greater than 20%, we narrowed down the markers a little bit more. And then there were no unique peaks to the negatives, and that was kind of what we wanted to see. And you can see that these molecules are pretty small. So we get to 83 to 225. 294 to 408 are probably too big for us to see, but we might be able to capture them on the breath. But we're really interested in the volatile organic compounds. So the average relative intensities of unique peaks common that are greater than 10% positive cases, without case 1 and negatives, were here. Here again, we see the average relative intensities for each one of the compounds. And so this tells us that we have small molecular markers, and these are in the aldehyde and ketone class that we could use for portable breath biopsy. So one of the ideas, of course, is just to develop an instrument that you can breathe into that will capture these aldehydes and ketones and re-release them into the mass spec for determining them. And that can be done in a myriad of ways. You can use membrane inlet mass spectrometry. You can use things like solid phase support structures. You can make chemically modified surfaces that grab onto the unique chemistry and then re-release them in the mass spec so that you can pre-concentrate them. But in every case, we're going to need to be able to pre-concentration because the dilution factor in the breath, as we all know, is pretty high. But we have a lot of these substrates nowadays. We have a whole class of 10X and Carbopac and Carboxyn, and we have membranes and Teflon and PDMS and all of these types of things that we can use. And then we can combine these things also for ways of capturing. But we have to know what the chemistry is first to be able to do that. So one of the ways that we can test this as a breath marker is using a flex stream system. Now this is the flex stream system that you see right here on the left of the instrument. And it is a 2-stage diluter with a permeation tube. So what you can do is you can create a standard of the compounds that you're interested in. You impregnate them into a Teflon tube. That Teflon tube is heated, released at a certain concentration, part per million to the high part per billion in the first stage, and then diluted again in the second stage here, down to the high part per trillion level. Now, the great thing about the flex stream is that the air carrier that you want to use can be anything that you want. So that you can make it breath, you can make it air, you can make it the atmosphere of Mars. You can do anything that you want with this breath system or air system to mimic the types of things that you're wanting to. So if you needed to add isoprene or different things into these compound mixes so that you can see if your instrument can tell the difference, this flex stream can do it. We've really loved having this. It's a great way to look at low concentration volatile organics to tell whether or not your instrument has the capability. And so, believe it or not, we created a 0.5 or 500 part per trillion concentration aldehyde and ketone mix. You can actually order an aldehyde and ketone mix with these 15 compounds that you see here, measuring from 29 M/Z to 134. And half of these are markers that we're actually looking for when we're looking for respiratory distress. And so we can use this to train our set on it. Okay, so we've built a couple of these portable instruments to date right now. So that you can grab the air compound and then determine it. We've connected it up to the flex stream and we started doing the preliminary tests. But you can also look in the high mass region down here, you can see them, and then start looking for people with concentrations. We can start seeing some small peaks of these aldehydes and ketones coming up in the background mix. And then of course this fragments, so we've got to be careful to identify the fragmenting properly. So here's the results of that. So look, we can see the acetaldehyde, we can isoaldehyde, formaldehyde, hexanals. We can see all of these things compound. They come out at different times because they're either released, they have different boiling points, or they have different diffusion coefficients. So they're actually coming back. So we get some real-time separation also with this technique. That's very interesting. Here you can see 4 more. Acetone being very particular. Acetone is very interesting, of course, because you can see that in diabetics. You can also see that in cancer patients. So acetone is one that comes up reproducibly. Acrolein 2 also is one. And then here are 4 more that we see pretty commonly. So it is possible to take all the way from the laboratory direct inject mass spectrometer, try to identify some of these low mass markers, then create the standards, create a technique to identify the standards, and now the next stage, of course, is to put this in clinical action. I will point to one of our colleagues here in SpecIR who've been doing work on the portable mass spectrometers, and I think they're going to do a poster during this session so that you'll be able to see some of the results that have occurred from actual samples of breath for COVID and non-COVID testing. Same with the opioid and the cannabinoid testing. I appreciate your time. Thank you very much.
So a few questions to come through. I'll read them out. There's 2 which are quite related, so I'll read both of them so you can answer those together. First is from Ahmed. From an analytical perspective, common challenge is the direct injection mass spec is ion suppression. Lack of separation would cause peak overlap. How did you find overcome these challenges? And related to that, from Mark, using ambient ionization, since you don't get extensive fragmentation molecules, how can you separate, identify, analyze with similar molecular ions?
Yeah, so that's a good question. I mean, on the bench scale, which is going to be a larger scale, yeah, we're using a linear ion trap on the front end. So you will have ion suppression effects. So we weren't really looking at being able to quantify on the breath. Now, the final instrument that you saw there is a linear quadrupole. And so, of course, that is quantitatively accurate, right? Because it's a scanning instrument. So so once you train sort of the interesting things on the linear quads, of course, you can do single ion monitoring if you have a right pre-concentrator where you're only isolating the specific molecules of interest. But if you're if you're looking for a plethora in training, you're absolutely right. I mean, but we have so much molecular species there that I was showing you before that, and ion suppression also, if you're looking at the same class of compounds, like in the case of the lipids when we showed up front in the single cell, we see reproducibility and that quantitative accuracy over and over again because the ions are, sort of in the same class. But yeah, it would become a problem if we were looking at the world and then trying to determine what the quantitative amount of each of the chemistry is that was present in that. There's a bigger problem too in the lysed cells of the COVID positive, COVID negative people, is that we don't know really how much of that organic load is actually there, right? How sick are they, or what is their viral load specifically that we're looking at on that. So quantitation wasn't as important on the front end as it will be, of course, in the biopsy side. So linear quad, if we're going to do mass spec as one of the detectors for breath biopsy, I think linear quad is going to have to be the one to do it. Yeah.
Great, thanks. There's another question, analytical setup. So regarding compound identification, how confident are you with the tentative identification of isobaric compounds in the absence of retention time?
Yeah, right. So we do MS. So we actually, that instrument's MS to the infinity. So we actually almost always do MS to the 3 in order to try to identify the compound specifically. So with liquid inject, direct inject, I've always been a big proponent of just trying to remove the LC off of the front of that, especially if you don't have a high number of compounds that you're looking for. But MS to the 3, we can easily identify which one of those constituents are there, of course, because each peak that you see there is a singly charged intact ion. Typically, most of them are thermally stable. We are running the front end at 150 C just so that we're a little bit colder than normal. Sometimes we run 200 or 250 C. We do get thermal breakdown, and we try to avoid that.
Excellent. Next question is from Anil. Would these VOCs in breath found in SARS-CoV-2 positive patients be found in other respiratory illnesses caused by other pathogens and viruses, etc.? The VOCs are created in the respiratory tract as a response to the incoming pathogen, so specificity is critical in creating a screening diagnostic test.
Yeah, so it's really interesting. I wouldn't have believed this initially until I read the BMC paper that came out in 2016 on the SPEMI. And they were looking at SARS and MERS and flu. It looks like that maybe flu viruses and coronaviruses are sort of in the same category as far as when the cells go apoptotic, they give sort of it looks like somewhere around 60% of the same markers, but there are 40% of these that are unique to the type of virus. And so that's hopeful. It's hard to answer that question, since our data sets are so small right now. But I'm really hopeful now, at least with the COVID outbreak, that we'll start to focus on flu. But there's 2 camps that I've noticed in the world around this. You have one, companies that say, well, we don't really care what they are. If they're sick, they're not coming in the building, right? So there's that side. And then the other side is the more diagnostic side. We're really saying, filtering people down and saying what's specifically wrong with them and where should they go. I think both are important, right? I think we'll have a lot of devices that will say sick, not sick, and then we'll have some devices that will specify. And I hope that the biochemical pathway for this breakdown, I mean, we kind of have an idea. It's the phospholipid breakdown of the membranes that are causing these aldehydes and ketones, but it would really be nice to know the exact pathway that each of the viruses are doing so we can see unique markers and build systems for unique markers. But
yeah, that's interesting. So the specificity may not be as problematic if you're just looking to screen out anyone that's got an effect. Yeah, that's
right. We talked with one company and they were like, yeah, we just don't want, if they're sick, we just want them to go see a physician. No, don't come to work that day. So, but we're also working with a group who are also looking at strep and other types of respiratory illnesses and seeing if there are, unique VOCs that identify, and we can tell, I mean, when we have strep, I mean, we all know, if the dogs, we can do it. I mean, when our kids are sick, I know they're sick, they smell bad. You can smell the markers on them. So there are things there. I'm very excited about breath biopsy really becoming a thing. I'm thankful for you guys putting this together. So it's fantastic.
So you mentioned some of those aldehydes and ketones. There's been a few papers published, some early stage biomarker discovery work, a few different groups, different analytical techniques. Are you seeing overlap with some of the similar compounds in that more recent reported literature in the COVID VOC work?
Yeah, we are on especially, I mean, specifically on the small molecules. The larger ones that of course the bigger instruments can see is they may sometimes be problematic. I guess we might be able to see them with something like an immunoassay type of, or an assay type device where we can capture some bigger molecules. But on the small side, yes, we were able to see the ones that I showed you on that were on that list. Half of the ones that were in that standard, it was kind of crazy. Sigma Aldrich actually had the standard ketone aldehyde standard, and half of those are known biomarkers for these states. And so we were, yes, yeah, that they didn't surprise us. And we kind of use the literature to help us identify the ones too. If we're going to do MS to the MSMS on these compounds, it's kind of hard to just do that blind. And so the literature guided us to the types of aldehydes and the ketones that we were and ended up identifying in that paper.
Great. Next question is from Veronica. Were your COVID positive patients in the study symptomatic, or did it also include asymptomatic individuals? If the latter, what was the fraction of asymptomatic? And could you observe differences in the spectrum of COVID positive patients based on severity of symptoms?
Yeah, so we had to do a filter down with our colleagues on this. We had to make sure that they were symptomatic patients. I mean, we're stuck in a really statistically weird world with this PCR analysis. And so if this, like I said in the talk, if this is a gold standard for us, the question is always going to be, how do you know if you do or you don't, right? In order to get these aldehydes and ketone markers, it's my opinion that the person's going to have to be symptomatic. I mean, right, their respiratory cells are going apoptotic, their body's trying to get rid of this chemistry. That means sneezing, coughing. So we asked in one of the questionnaires is, you know, what were their symptoms? And so the results that you see there are people that have fever, that have, you know, high viral load. And so they were definitely symptomatic folks. And that was just to help us with the statistical analysis. We didn't want to rely on PCR alone to get our results because of the fail rate.
But does that limit the potential utility? So from that hypothesis there, if there's unlikely to be VOCs produced from the asymptomatic patients, we know the virus does get transmitted by asymptomatic patients.
Yeah, they do. I mean, transmission for asymptomatic patient would be a little bit different than symptomatic because they're not coughing and sneezing, right? So, but we felt it more important to identify the chemistry up front and have more you know, idea of what these exact VOCs were when the cells went apoptotic. And we weren't confident enough that if you had an asymptomatic patient that they were positive or negative. So that's up front. Of course, it's not so easy to keep reproducing this experiment, as you can imagine. You know, COVID's a hard experiment to do in the lab. So yeah, but
yeah. Great. Next question was, what do you think is the reason for the high organic load in the COVID positive samples? Is it related just to the human body response or also to the virus biological material itself? Did you evaluate this?
Yeah, we didn't. I mean, we went after the specific type of chemistry, right? Doing MRMs on that. So the data is farmable for us from now until forever. So that's kind of nice. So we have all the data sitting there. If we want to look at another class of compounds, if we want to look at fatty acids and the lipidome, or even if we want to start looking at, you know, some other ones like, I don't know, cholesterols or different hormones, they're there. So it is at least for us, it's a farmable spectrum. It's a good question. We started there, but we haven't gone down the rabbit hole of looking at what else is systemically going on there. I mean, like, because you're going to have other processes like inflammation and different things like that that are going to occur. And there may be some markers that we can identify from that, but we just haven't gotten that far yet.
Got it. And then the schematic for the portable mass spec, where you have things like membrane inlet, it looked like the samples coming from the enclosed chamber. So you'd mentioned dilution as a potential problem there. So need to pre-concentrate. You could get some selectivity from the chemistry on the inlet of pre-concentration as well. But would you worry again about the contribution of the background? Yeah, we know there's aldehydes and ketones floating about just in the ambient. So curious to see if you think that's likely to be a problem here.
Yeah, I mean, that's our big question right now, because where are we going to put this, right? If we put this in the front of a corporation, of course, the air, the surrounding air is going to be a lot different than if we put this in a hospital or in some more of the settings. And so, yeah, that will have to be done for sure. Yeah. We always like the aromatics when we're talking about drug detection, because you know, they tend to be less likely to be in our air if you're not around diesel fuel and that kind of stuff. But the aldehydes and the ketones, you're absolutely right. Background will have to be taken and subtracted. Now, in the membrane inlet case, you'll see the spike up. So that'll be the subtraction from the background. So in the membrane inlet case, when you see the rise in the peak from the diffusion coefficient, that's going to be coming from the sample, not from the background, because the background's permeating the membrane all the time.
Yeah.
For the capture device, that is a problem, because we're capturing everything, even the ambient, and then we're releasing it. So when we see that release, the release is not coming from the person breathing, it's coming from the instrument popping it off of the substrate. We get better pre-concentration with that and better selectivity with that device, but yes, those experiments need to be done. Yeah.
Excellent. I think we have time for a couple more. So one question for the portable mass spec is very promising. Do you think similar performance could be attained with portable ion mobility or differential mobility spectrometers?
Yeah, so my dissertation thesis is on ion mobility coupled to mass spec. So I walk both worlds. IMS is great. It's small system, very portable. You don't need to pump and vacuum systems like you do in mass spec, but its selectivity is not quite as good. So the peak capacity of our system is roughly around 300, where of course the peak capacity of ion mobility is somewhere around 20. I do think so. I mean, I think when most of these front ends, if you're building front ends that are highly selectable for the specific types of chemistry that you're interested in, then you want the detectors to get smaller and smaller. Mass spec is really, and in our group, we really do a top-down approach. So the mass specs that I'm showing you have benchtop quality. There are other mass spec groups out there in the world, of course, like 908 devices that do bottom up, and they can make really small devices, but their peak capacities are awfully low. And we try to maintain our devices that when we show you a mass spectrum, it still looks like a mass spectrum to a mass spectrometrist. And so that's why our systems are a little bit bigger. We hope the vacuum technology gets better and better, but in the meantime, IMS is a good ion detection device. So,
do you think pre-separation, GC pre-separation would be required then, just to account for that limited peak capacity and also deal with issues around what's going to get ionized if you're using, nickel or UV source or whatever the case may be?
Yeah, absolutely. I think in both cases, you'll have to have some sort of separations. Now, the interesting idea that we've been working on in our group is called earth-based separations. And so when you looked at those last few slides, what you saw was that the diffusion coefficients going through the different membranes or even going through the air is not the same. And because of that, that's sort of a pre-separation technique. So as you see one peak coming in and you start seeing the ionization signature or the fragment signature of that specific molecule, you can start tracking that and subtracting it from when the next molecule comes in, and so forth and so on. And we started doing these algorithms where we can take multiple components and we let the diffusion coefficient, because these devices are fast, right? We're sampling at 10 milliseconds, 20 milliseconds per scan. So we have the capability of being able to capture these devices in an earth-based separation mechanism. And so we think that that's important to look at, because that's just another factor for us. What does the diffusion coefficient look like, and can that tell us something about what the molecule is also?
Excellent. And then the last question is, could you estimate the specificity of the biomarkers from the point of view of separation of COVID from healthies?
Okay, say that one more time.
Could you estimate the specificity for separation of COVID?
Oh, I mean, that's yeah. So I think that, yeah, that's a person, that's a clinician, sure, right? So yeah, because specificity and sensitivity, you guys are you have these equations for that. I don't know that. I could calculate it. I'm sorry, I should have. It's definitely a question that the FDA asks us. Of course, if you want emergency use authorization, you have to turn all this in. So I know the guys that inspect IR know what that is, and John and Tim will probably be able to answer it for y'all. But yeah,
No.
sorry. I wish
No worries, that's great, thanks. And then the very last one, what's the level of development of the portable mass spec for direct clinical applications?
Yeah, so the Inspect IR guys have done pretty well. They've identified engineering firms, and they've got the first handful of devices made. So the ones that I showed you in mine are, of course, my Frankenstein devices that I make in the lab, but they're not very clean. But Inspect IR's done that. Yeah, so you can see those now. And of course, FDA emergency use authorization over here in the States is even though almost 75% of all the applications for breath biopsy does not require any FDA, everybody asks for it. So it's, you know, when is this going to be FDA approved? So we're kind of in a catch-22 right now. But
Excellent. Well, thank you again for fantastic talk. And I know it's very early for you, so to be able to go and get that coffee and get stuck in.
Yeah, that'll be good. I get my day started really early. Thank you.
Thanks again. Have a great rest of your day. Enjoy the rest of the conference.
All right, I really appreciate it.